Let's continue our talk about active transport. First of all, remind me, what is active transport? Pause me, think about it, and then come back when you have the answer. It's movement of molecules from low to high against their concentration gradient using energy. What was the kind of active transport that we learned about already? It was movement with a carrier protein. Today we're going to look at another form of active transport. So again, takes energy, sledding uphill, going from low to high against the concentration gradient. We're going to look at endocytosis and exocytosis, which are types of movement using vesicles. In cells before, we learned about the organelles, and vesicles, remember, like little taxi cabs of the cells that move things around. Endocytosis and exocytosis are opposite processes from each other. Endo means in. So endocytosis is bringing things into cells by forming a bubble. The substance pushes into the cell membrane, which causes a little bubble to form and pinch off and move inside the cell so it can go and be transported wherever it needs to go. This is active transport. It takes energy to happen. We have two kinds of endocytosis. Phagocytosis means cell eating, and this is transporting whole cells or particles inside the cell. If you look at this YouTube video, we're going to see an amoeba eating a paramecium. So this blob here is an amoeba. It's surrounding a protist called paramecium, bringing it into its body. That was endocytosis that you just saw happen right there. So now the paramecium are both inside the amoeba, and then they're going to get digested, and they're freaking out because basically they're being destroyed. Penocytosis is a word that means cell drinking, and this would be bringing liquids in. So again, these are both kinds of endocytosis. Phagocytosis is bringing in solid molecules. Penocytosis is bringing in liquid molecules. Same way, they push into the cell membrane, which causes a bubble to form and bring whatever material it is inside the cell. The opposite process is exocytosis. It's basically endocytosis in reverse. So it's sending a large molecule out through a vesicle. The vesicle starts inside the cell, merges with the plasma membrane, and then gets kicked out. This animation is going to show exactly what's happening. So as I step through, here comes a bacterium that is going to push in towards the plasma membrane. This is phagocytosis. It's cell eating because it's a solid material. Keeps pushing forwards until a little bubble forms and then enters the cell. Here's penocytosis, a liquid. Pushes in, pinches off, enters the cell. This is what's happening at the cell membrane level. You don't need to worry about that. And basically, at the end here, we have exocytosis, where we're sending something out by merging it with the cell membrane, and then it gets kicked out of the cell. I could play this in reverse, and it looks like endocytosis, exocytosis. Why is all this stuff important? Why do we care about transport? Well, transport's happening in your cells all the time in a variety of ways, and it's what's keeping you alive right now. So for example, in digestion, you eat lunch, your body's going to break down that food into amino acids, into nucleotides, into glucose, and you need to get all that stuff from your blood to your cells. And it's going to diffuse in and out of your cells from high to low concentration. Water's going to move through osmosis, certain things will go through active transport, just to get where it needs to go. In terms of respiration, carbon dioxide and oxygen are carried in your blood and travel in and out of your cells by diffusion. So in your lungs, oxygen is high when you breathe in, and the blood coming in is low in oxygen. So the air is going to diffuse from your lungs into your blood. Your blood is high in carbon dioxide because it's carrying what it picked up from your muscles, and then it's going to bring that to your lungs, which are low in carbon dioxide. Molecules are going to diffuse into your lungs. You breathe carbon dioxide out. Another reason why it's important is getting rid of wastes. Osmosis and diffusion are used to help concentrate urine, so then you can pee things out and get rid of them. If you're going into nursing, this is super important because medications, IVs, rely really heavily on osmosis and diffusion. If you don't follow what's going to happen, you could end up hurting your patient more than helping them. In terms of farming, Fertilizer, you saw that in the case study that you read. Treating soil and crops, treating animals. If you're not careful with medicines and fertilizers and the principles of diffusion and osmosis, you can actually end up, again, hurting things more than helping them. Next steps, you have a worksheet to complete. Grab it from the crate. When you're done, you're going to turn that in. Your job is to create a concept map for the information in this unit. By now, you should have a good handle on how to do it. So you're going to complete this on a separate sheet of paper, make sure your name is on it, and turn it in. This is reviewing everything that you learned about in this unit. If you're still kind of shaky and stuck on making a concept map, call me over and I'll help you, and there's probably other people who feel the same way, so we can get a small group going and make a concept map for this unit if you need help. You may work with one partner if you are at this point, and make one map for the two of you. No more than partners though. Do this on a blank sheet of paper, no lines. Don't forget to put your names on it, turn it in. And if I have any questions, I'll be seeing you to help clarify what's going on. Lastly, see the last page of your notes for your test and how it's going to work for this section, because it's not exactly a normal test. So check on there, and if you have questions about it, let me know.